Hello, everyone. The topic today is about proteins. Proteins are large biomolecules or macromolecules that comprise one or more long chains of amino acid residues. They play many critical roles in the body. Many types of proteins exist, and they perform a variety of functions, including the following. As enzyme, they are the most varied and highly specialized proteins with catalytic activity. Structural proteins. These proteins aid in the strengthening and protecting biological structures. Transport. They are involved in the transport of ions or molecules. Nutrient. These proteins provide nutrition to growing embryos and store ions. Contractile. These proteins function in the contractile system. Defense. Such as antibodies, these proteins defend against other organisms. Regulatory. They regulate cellular or metabolic activities. And the toxic proteins. These proteins hydrolyze or degrade enzymes. Example is snake venom. Although a wide variety of proteins exist, they all have basically the same structure. They are chains of amino acids, and its name implies an amino acid is an organic compound containing an amino group and a carboxyl group connected to the alpha carbon. This is the carbon that is connected also to four other groups, and therefore it is a chiral carbon. Organic chemists can synthesize many thousands of amino acids, but nature is much more restrictive and uses 20 common amino acids to make up proteins. This slide shows you the stereochemistry of alanine and glyceraldehyde. This is D-glyceraldehyde and L-glyceraldehyde. Take note of the orientation of the hydroxyl group, hydroxyl group. Hydroxyl group to the right is D, hydroxyl group to the left is L. Now, in the case of amino acid alanine, if your amino group is to the right, this is your D-alanine. And if your amino group is to the left, it is your L-alanine. The amino acids found in proteins have the same chirality as L-glyceraldehyde, which is opposite to that of D-glyceraldehyde. All but one of the 20 amino acids fit the general formula of the alpha amino acid. This is an alpha amino acid in which the amino group is linked to the carbon next to the carboxyl carbon. So we have here a chiral carbon that is bonded to four different groups. These are the amino group, R group, carboxyl group, and a hydrogen. Amino acids are the best known examples of two-veter ions. Two-veter ions are compounds that have a positive charge on one atom and a negative charge on another. The term two-veter ion comes from the German word "Zuwitter", meaning hybrid. Amino acids are two-veter ions, not only in water solution, but also in the solid state. They are therefore ionic compounds, that is, internal salts. Unionized amino acid molecules do not actually exist in any form. The fact that amino acids are two-veter ions explains their physical properties. All of them are solids with high melting point, just as we could expect of ionic compounds. The 20 common amino acids are also fairly soluble in water, as ionic compounds generally are. If they had no charges, we would expect only the smaller ones to be soluble. Let us define isoelectric point or isoelectric pH. This is a pH at which a sample of amino acids or protein has an equal number of positive and negative charges. This slide shows you the list of the 20 amino acids in alphabetical order. This column is their three-letter abbreviation. Next column is the single letter abbreviation. And the last column is isoelectric point or isoelectric pH. Amino acids are grouped according to the characteristic of their R group. So let us take a look at their R group: CH3, CH3CH3 for valine. This is methionine, CH2, SCH3. Leucine, take note, it is also composed of hydrocarbons, likewise hydrocarbon. In the case of proline, its R group consists of this. So it's a CH2, CH2, CH2. The only difference is that its R group is connected to the amino group. So this group of amino acid is a non-polar, is classified as non-polar amino acids because the R groups are non-polar. Let us take a look at the R group of these amino acids. In serine, it has an OH group, threonine, another OH group, tyrosine, OH group, cysteine, SH group. Tryptophan, it has an NH. Asparagine, it has an NH2 and C double bond O. Glutamine, NH2 and C double bond O. So this group is polar neutral amino acid. But take a look at glycine. Its R group is a hydrogen, another hydrogen. In some books, this is placed under nonpolar, nonpolar amino acids, but other uh, books contend that since hydrogen is a small atom, then it can be easily polarized. Kaya andyan siya sa polar neutral group. Now, let us examine the R group of these amino acids. Its R group contains a carboxyl group. Carboxyl group. This is aspartic acid. This is glutamic acid. It has a carboxyl group in its R group. So, what do we know about carboxyl group? Carboxylic acid. So, these amino acids are considered polar acidic amino acid. It has an acidic group in its R group. What can we notice about the R groups of these amino acids? Maraming amines. amine, amines. Okay, and what do we know about amines? They are basic. So this group of amino acid is called basic polar amino acids. Since the side chains are the only differences among the amino acids, ultimately, the functions of the amino acids and their polymers, the proteins, are determined by these side chains. For example, one of the 20 amino acids in the previous table has a chemical property not shared by any of the others. This amino acid is cysteine. It has an SH group. Cysteine can easily be dimerized by many mild oxidizing agents. The dimer of cysteine, which is called cysteine, can in turn be fairly easily reduced to give two molecules of cysteine. As we shall see, the presence of cysteine has important consequences for the chemical structure and shape of protein molecules of which it is part. The bond shown, okay, so this is the bond, is called a disulfide bond or disulfide linkage. Several of the amino acids have acidic or basic properties. Two amino acids, glutamic acid and aspartic acid, have carboxyl groups in their side chain. So we refer to this carboxyl groups. This is their side chain. It has a carboxyl group. In addition to the carboxyl group dito, okay, the carboxyl group that is present in all amino acids. A carboxyl group can lose a proton, forming the corresponding carboxylate anion. In this case, the 2 amino acid has an extra negative charge if they are placed in neutral pH. 3 amino acids, histidine, lysine, and arginine, have basic side chains. Take note of their side chain: positively charged, positively charged, positively charged. This is these side chains become positively charged at near or neutral pH. So this is in contrast to the acidic amino acids where the charge of the R group at neutral pH or near or neutral pH is negatively charged. The amino acids phenylalanine, tryptophan, and tyrosine have aromatic rings in their side chains. So ito yung aromatic ring, this is the aromatic ring, and this is the aromatic ring. They are important for a number of reasons. As a matter of practicality, these amino acids allow us to locate and measure proteins because the aromatic rings absorb strongly at a wavelength of around 280 nanometers and can be detected using a spectrophotometer. Tryptophan, for instance, is very important physiologically because it is a key precursor to neurotransmitters. Neurotransmitters are substances involved in the transmission of nerve impulses. Tryptophan is converted to serotonin, or more properly called 5-hydroxytryptamine, which has a calming effect. Very low level of serotonin are associated with depression, whereas extremely high levels produce a manic state. Manic depressive schizophrenia, also called bipolar disorder, can be managed by controlling the levels of serotonin and its further metabolites. Tyrosine, also which can also be derived from phenylalanine, is converted to the neurotransmitter class called catecholamines, which includes epinephrine, commonly known as adrenaline. Some organisms, including bacteria, can synthesize all the amino acids that they need. Other species, including humans, must obtain some amino acids from dietary sources. The essential amino acids in human nutrition are listed in this table. The body can synthesize some of these amino acids, but not in sufficient quantities for its needs, especially in the case of growing children, particularly children's requirement for arginine and histidine. This is the end of this presentation.